welcome back to Biotech. Then we will study Annexure from the second chapter. In the last class we studied about the cell, the basic unit of life, with full chapter in one class with important bits. We have underlined each and every important bit we have underlined with a red marker. So whatever it is underlined with red marker, that is very important. It is asked in examination several times. So now we shall study annexure. So for every chapter there might be some annexure after the lesson. This annexure is also very important. Annexure means which is related to the lesson. Suppose in the second chapter we studied about the cell, the basic unit of life. How cell is observed? The cell is observed through the microscope. First time the Robert Hooke observed the cell with the help of the microscope. So what type of microscope he has used? In earlier time, how many different types of microscopes were invented? So that we are going to study. Then how can we prepare the slide? Or what is the use of microscope? How can we observe the objects through the microscopes? That we will study today with small topic, but there will be plenty of information with most important points. Let us see here. First one, in annexure, see, we shall study about the microscopes. Here we are going to study about microscope. So what type of microscopes were there? In the earlier time, major improvements were made in the microscope from around 400 years ago. That means before the 400 years ago, we do not know about the microorganisms or microbial world. But whenever the microscope is invented into the world, so whenever we started the observing different types of microorganisms with the help of microscope, so we came to know all the things. There were microorganisms like bacteria, algae, fungi, protozoans, microarthropods, viruses, etc. So all the knowledge we have gained after the discovery of microscope, that was from the where 400 years ago. So since 400 years ago the microscope is invented, and this microbial world knowledge came into the use into the world. This is the most important word. And next one, in the early days there were used only single lens microscope. The first microscope is with the single lens microscope. So single lens microscope is generally, even the Robert Hooke or Anthony van Leeuwenhoek is also, Anthony van Leeuwenhoek, when he observed the microorganisms through the microscope, he used the single lens microscope. That single lens microscope can magnify up to 300 times. Yesterday we discussed in the class, when Anthony van Leeuwenhoek observed the microorganisms in the pond water. So when Anthony van Leeuwenhoek observed the microorganisms in the pond water through microscope, that microscope is with single lens of microscope. That microscope can magnify 300 times more. So here also, in the early days, single lens microscope is used. This single lens microscope is also called as a simple microscope. Only ఒక్క సింగిల్ లెన్స్ మైక్రోస్కోప్ కనుక ఉంటే ఏమని పిలుస్తాం? దాన్ని సింపుల్ మైక్రోస్కోప్ అని పిలుస్తాం. వెరీ వెరీ ఇంపార్టెంట్. వాట్ ఈజ్ సింపుల్ మైక్రోస్కోప్? సింపుల్ మైక్రోస్కోప్ మీన్స్ if the microscope is used with only one lens, that is called simple microscope. Then next one, later there were several lenses were made or prepared. More than one lens, or 2, 3, 4, so like that there are several lenses were made. So that several lenses were made and introduced into the world. Suppose if the more than one lens is used in the microscope, that microscope is called as a compound microscope. Very very important. What is the difference between the simple microscope and compound microscope? Simple microscope, in the simple microscope there is only one lens, but in the compound microscope more than one lens is used. That is called compound microscope. Even the Robert Hooke, when he observed the non-living cell, Robert Hooke, he observed the non-living cell. That means he took the oak tree plant bark and he kept under the microscope and he observed. When he observed the oak tree plant under the microscope, he had seen there will be several little rooms. This each and every little room is called as a cell. So even the Robert Hooke, what type of microscope he used to observe the oak tree plant bark? Compound microscope is used. Very, very important. So Robert Hooke, when he observed the oak tree plant bark, what type of microscope is used? So compound microscope. That is not the simple microscope, but Robert Hooke, what he used the microscope is compound microscope. Compound microscope means more than one lenses are used for the compound microscope. Suppose examples here. The lenses maximum usually in the compound microscope there is 4 into 10 into 40 into 100 into powers of lenses will be used. So like this, more than one lens are, if the more than one lenses are used to observe the things through the microscope, that such type of microscope is called as compound microscope. That is about simple microscope and compound microscope. This compound microscope is developed by the Jacques Janssen in the year of 1595. Very, very most important bit. Who invented the compound microscope? Compound microscope is invented by the Jacques Janssen in the year of 1595. It is specifically mentioned in the textbook. That is what in the year of 1595 Jacques Janssen developed the compound microscope. Okay, that is about use of microscope. So how can we use the microscope? In the 6th class science we studied about the parts of the microscope. What are the different parts of microscope and how can we use the usage of microscope in the 6th class science. But here we shall see some important points regarding the usage of microscope. First, the lens, the eyepiece. Generally we shall see the eyepiece are lenses. That lenses should be clean with a dry cloth and soft cloth. That lenses should be cleaned. And next one, the microscope, whenever we are observing the objects, that microscope should be kept in the light focusing area, where there will be the light focusing area, then there will be, we shall use the microscope so that we can observe the things very clearly. So light focusing area is must be needed for observation of any material through the microscope. And next, usually in the microscope there were 3 or 4 lenses of powers, that is 4 into 10 into 40 into 100 into powers. This is what about compound microscope. In the compound microscope more than one lenses are used. That is very important. This is also important. Next, preparation of microscope slide. Whenever we are going to prepare a slide, here first the slide measurements, it should be 3 centimeter width, 8 centimeter length and 2 millimeter thickness. This is the measurement of the slide. So 2 millimeter thick and 3 centimeter into 8 centimeter rectangle glass slide is taken. So the glass slide measurements are specifically given in the textbook. That is what 2 millimeter thickness and 3 centimeter into 8 centimeter rectangle glass slide is to be taken. And then this glass slide, it is generally used to prepare any temporary mount or permanent slide. Suppose if you want to prepare a dicotyledon stem or monocotyledon stem, or for the observation of nucleus of onion peel cells. So like this we shall prepare the different types of permanent slides. When we prepare the permanent slides, so this type of slides we should be taken. That slide should have 2 millimeter thickness and 3 into 8 centimeter rectangle glass slide is to be taken. Besides that, we also take some cover slip, glycerin are needed. Generally cover slip ఏం చేస్తుందంటే, కవర్ స్లిప్ అనేది ఏదైతే మనం స్లైడ్ తయారు చేస్తున్నామో ఆ స్లైడ్ తయారు చేసిన తర్వాత అది డ్రై అయిపోకుండా దాని మీద ఆ మెటీరియల్ పైన గ్లిజరిన్ వేస్తాం. గ్లిజరిన్ వేస్తే అది డ్రై అయిపోదు. దానిపైన కవర్ స్లిప్ వేస్తే ఈ కవర్ స్లిప్ అనేది మనం అక్కడ లైట్ ఫోకసింగ్ చేస్తున్నప్పుడు ఈ స్లైడ్ తయారు చేసిన మెటీరియల్ అనేది ఆ లెన్సెస్కి అంటిపోకుండా ఉండడానికి ఉపయోగపడుతుంది. దట్స్ వై వీ షల్ యూజ్ ద కవర్ స్లిప్. సో కవర్ స్లిప్ అండ్ గ్లిజరిన్ ఈజ్ వెరీ ఇంపార్టెంట్. గ్లిజరిన్ కెన్ ప్రివెంట్ ద డ్రైనెస్ ఆఫ్ ద స్లైడ్. అట్ ద సేమ్ టైం కవర్ స్లిప్ ఏం చేస్తుంది అంటే, కవర్ స్లిప్ అనేది స్లైడ్ మనం అబ్జర్వ్ చేస్తున్న మైక్రోస్కోప్కి ఆ లెన్సెస్కి అతుక్కపోకుండా మెటీరియల్ అనేది ప్రొటెక్ట్ చేస్తుంది. నెక్స్ట్, ఆబ్జెక్ట్ షుడ్ బి కట్. ఏదైతే మనం ఆబ్జెక్ట్ని మైక్రోస్కోప్ కింద పెట్టి చూడాలనుకుంటున్నామో, ఏదైతే స్లైడ్ తయారు చేయాలనుకుంటున్నామో ఆ ఆబ్జెక్ట్ని ఎంత మొత్తంలో కట్ చేయాలంటే 0.5 millimeter థిక్నెస్లో కట్ చేయాలి. సపోజ్ ఎగ్జాంపుల్, ఒక మెటీరియల్ ఏదైనా ఒక సపోజ్ టీఎస్ ఆఫ్ స్టెమ్ అంటే ట్రాన్స్ఫర్ సెక్షన్ ఆఫ్ డైకాటిలిడన్ స్టెమ్ కనుక కట్ చేసి చూడాలనుకుంటే ఆ స్టెమ్ని ఎంతలో కట్ చేయాలంటే 0.5 millimeter ఆర్ లెస్ థిన్ సెక్షన్. ఇంత థిన్ సెక్షన్లో కట్ చేస్తేనే దెన్ వీ కెన్ అబ్జర్వ్ ద మెటీరియల్ అండర్ ద మైక్రోస్కోప్. సో దిస్ ఈజ్ ఆల్సో వెరీ ఇంపార్టెంట్. Cut the object nearly 0.5 millimeter or less thin section. That is the very important. Next, to observe clearly, staining is also very important. So staining means coloring. That staining should be done with either iodine or saffron or fast green or any other chemical dyes will be used to staining that objects. So staining చేయడం వల్ల ఏమవుతుందంటే చాలా క్లియర్గా మనకు మైక్రోస్కోప్ కింద ఆ ఆబ్జెక్ట్ కనబడుతుంది. Suppose example, if you want to observe the nucleus in the onion peel cells, so we must stain that onion peel cells with saffron. Then saffron వేస్తేనే ఆనియన్ పీల్ సెల్స్లో ఉన్న న్యూక్లియస్ అనేది కలర్ ఆబ్జెక్ట్గా మనకు దాంట్లో క్లియర్గా కనబడుతుంది. కాబట్టి సాఫ్రిన్ ఆర్ అయోడిన్ ఆర్ ఫాస్ట్ గ్రీన్ ఆర్ ఎనీ అదర్ కెమికల్ డై షుడ్ బి యూస్డ్. సో దట్ స్టెయినింగ్ ఈజ్ మస్ట్ బి used. Staining is compulsory. That staining will be done with the different types of chemicals like iodine, saffron, fast green or other any chemical dye. కాబట్టి ఎగ్జామ్లో మనకు ఈ క్రింది వాటిలో స్టెయినింగ్లో ఉపయోగించని కెమికల్ మెటీరియల్ ఏంటి అని అడుగుతారు. కాబట్టి iodine is used, saffron is used, fast green is used, then other chemical dye. These four we shall remember. Then to make the saffron, మరి సాఫ్రిన్ లేదా ఈ స్టెయినింగ్కి కనుక యూజ్ చేసే అంటే సాఫ్రిన్ ఎలా తయారు చేస్తాం అంటే saffron should be dissolved in a 100 ml of water. So 1/4 teaspoon of saffron. ఒకవేళ సాఫ్రిన్ కనుక తయారు చేయాలనుకుంటే మనం 1/4 teaspoon of saffron should be dissolved in 100 ml water. Then it becomes saffron solution. So like this we shall prepare the saffron solution. So saffron is used to staining the objects to see under the microscope very clearly. So this is all about the annexure. In this annexure, very most important things that we should remember. Microscopes were earlierly 400 years ago back, they were invented into the world. Then in the earlier days, the microscopes were generally simple microscopes. Simple microscopes means there will be only single lens will be used. Then after there were several lenses are invented. These several lenses, more than one lens if they are used, that is called compound microscope. Even Robert Hooke also used the compound microscope. Generally these lenses are either 4 into 10 into 40 into or 100 into powers. Next, when you are using the microscope, we should clearly clean the lenses of the microscope, and that microscope should keep it in the light focusing area. And next one, preparation of microscopic slide. When you prepare the microscopic slide, that microscopic slide should have some particular measurements, that is 2 millimeter thickness and 3 into 8 centimeter rectangle glass slide is needed. And while preparing the permanent slide, we should take the cover slip and glycerin. So glycerin can prevent the dryness of the slide, whereas cover slip is preventing to attach the material, slide material. స్లైడ్ పైన ఉన్న మెటీరియల్ అనేది దేనిపైనకి మైక్రోస్కోప్కి అతుక్కుపోకుండా మనకు కవర్ స్లిప్ అనేది ప్రొటెక్ట్ చేస్తుంది. అంటే స్లైడ్ పైన ఉన్న మెటీరియల్కి ఇటు మైక్రోస్కోప్కి మధ్యలో కవర్ స్లిప్ అనేది ఉండడం వల్ల మనం పర్మనెంట్గా తయారు చేసిన స్లైడ్ అనేది దానికి అతుక్కుపోకుండా కాపాడుతుంది. సో నెక్స్ట్ వన్, స్టెయినింగ్ ఈజ్ ఆల్సో వెరీ ఇంపార్టెంట్. Staining should be done with iodine, saffron or fast green or any other chemicals. And to prepare the saffron, compulsory the saffron 1/4 teaspoon of saffron should be dissolved in 100 ml water. So this is all about the annexure regarding the microscopic preparation of slide. So to observe the microscopes. So for more updates, then please subscribe, and we'll see some other topic in the next class or in coming class. Thank you.